cattle. We've got our own breed called the Drought Master, for instance. Anyway, so they brought me back to the lab to do this project, which I did, and wrote a master's thesis from it. And also I was doing diagnostic pathology, and I was, and then they sent me off to learn some virology because they knew I was interested in viruses. But about that time, I was getting a bit restless. I'd spent about 4 years there and wanted to move on and do a PhD. And normally, I looked around and I've discovered that the Mordun Research Institute in Edinburgh, which is a sheep disease place, worked originally with Scrapie, you could go and work there and do a PhD. And I was about to write to them when I saw a job advertised. This is before the internet. The jobs were all advertised in the back of the journal Nature. And there was only one Nature, not 10 of them or something, just Nature. And I wrote off, and there was this job advertised for a veterinary neuropathologist. Must be very experienced, must be trained in neuropath and all the rest of it. I wrote off and said, well, I don't know any veterinary neuropathology, but I've heard I can do a PhD at the University of Edinburgh while I'm working there. And if you don't get anyone for the job, I'd be a bit interested. And so they wrote back and said, come immediately, because I was the only applicant. And so I then went to Edinburgh in Scotland from Queensland, which Edinburgh was wonderful. The sun never shines there, or didn't before global warming. So I could go outside all day and not get sunburnt. I've got a very fair skin, so I really love the place. And it's always raining, bleak. It's very bleak, actually. They have their own word, dreek, to describe it. So I went there and I did a PhD in neuropathology while I was employed by the Scottish agriculture department, and was working full-time as a senior veterinary pathologist. And then completed the degree part-time as an external student. I used to go, my boss was a very good pathologist, and he taught me neuropath. I did a PhD on louping ill encephalitis of sheep. It's a tick-borne encephalitis from the point of view of pathology. And I worked with another young colleague who was also doing his PhD. So I wrote my PhD on the pathology. He wrote his PhD on the virology and immunology of the same sheep as we were doing these experiments. And it was fun. And I was doing a lot of postmortems, also postmorteming horses, doing neuropath. There's a lot of issues in neuropathology. I think when you've taken the spinal cord out of a horse, you really know you're confronting reality, because it's a pretty hard work, quite frankly. And finished my PhD, a lot of electron microscopy and pathology and some functional stuff as well. Wrote some papers and was doing quite reasonably well in the veterinary world. Then my wife and I had married before we left Australia. We were being offered a job back in Australia in the big government health organisation, the Commonwealth Scientific Industrial Research Organisation, which at that stage was very oriented towards animal health. And I was being offered a job as a principal veterinary researcher working on viruses and immunity in domestic animal species. I would have ended my career if I'd taken that job at the big animal health laboratory, which is the only high security animal health laboratory in this part of the world. We could handle foot and mouth, or we did handle Nipah virus and Hendra virus and all those sort of things in that. And we could actually infect an alpaca or even a camel, though they didn't really want to do that, and handle that under level 4 security conditions. It's a very sophisticated place. But I talked to the guys who were going to give me the job. There were plenty of jobs around, said, I wanted to learn something about cell-mediated immunity. And I knew there was a very good group working in the John Curtin School of Medical Research in Canberra, Australia, on T cells. So I said, do you mind if I can find some money somewhere? I go there for a couple of years. And so I did. They were happy about that. They were pretty relaxed. And so I went there, and I was kind of an odd fish, because I was already lined up to go and work in the CSIRO, and here I was working on a junior sort of thing. They left me alone. I ended up in the lab by myself with a technician. They were tiny labs. It was the old days when scientists worked with a student or two and a technician, and they didn't have groups or 8 or 12 or so forth around them. And I started to do experiments in mice, And then Rolf Zinkernagel, a young MD from Switzerland, came to work with someone else there, but they didn't have much room in the lab, so they put him in with me. We started to talk, we started to do experiments, and we made the discovery that won the Nobel Prize 20 years later. It was a chance discovery, totally chance. And we wrote this up. It was published in Nature. There was a format in Nature called the Letter to Nature, which had 3 or 4 articles of presentation at about 1,000 to 1,500 words. And we developed a whole theory around how the immune system works from our experiments. And we won the Nobel Prize many years later for that theory and those experiments that changed our whole understanding of how cell-mediated immunity works. And that was the first actual Nobel Prize ever for cell-mediated immunity. We've just had one to 2018, we had one to Jim Allison and to Suku Honjo for the clearing of cancer cells by turning them back on again with turning off those molecules that inhibit them. So basically, that transformed me from being a totally unknown person, completely unknown, to suddenly being notorious. And because nobody quite understood the experiments, we turned transplantation around backwards. We said it's not about rejecting tissues, it's about presenting changes in our own cells so that our own cells can monitor them. It's about immunological surveillance. So we turned immunology on its head. And a lot of people were very upset by that. People who are very established scientists, very famous, don't really want 2 unknown young guys to come along and show them that they really missed it. They missed the point. So we weren't universally loved. And also the theory we proposed, which we couldn't prove at that time because it was very primitive. We had no monoclonal antibodies, no recombinant DNA technology, no PCR, all the things that we so rely on now and allow us to do such incredible science. We had none of that. So you could speculate wildly. And that suited me very well because I love to speculate. Now, of course, if you put speculation in a paper to Nature, they tell you to go away and do the experiment. And you can. But fortunately, at that time, you couldn't do the experiment. So you could speculate, which was fun. And so we wrote a lot of papers, and I left because there wasn't, I didn't go to the CSIRO. I went to the Wistar Institute in Philadelphia. From there, I was teaching in the University of Pennsylvania. Wonderful, wonderful research medical school. Also very interesting because I was part of what's called the Immunology Graduate Group. They have this structure where anyone learning immunology around the University of Pennsylvania, all the associated research institutes that are really quite independent, like the Wistar Institute or Fox Chase Cancer Center, they were all part of the University of Pennsylvania Immunology Graduate Program. That included the vet school. They have a wonderful vet school at Penn, and it's right next door. The urban campus is right next door to the medical school. So it really was a very good experience. And then I became a medical researcher. So I went overnight from being a vet to being an MD, a mouse doctor. And that's how I spent my research career, pretty much studying immune responses in mice, which of course, because of the genetically defined systems there, firstly inbreds and back crossing to isolate the transplant molecules. And then of course, all the transgenic and knockout mice and so forth. This was a wonderful experimental system for understanding the pathogenesis of virus infections. So that's what I've worked on. Apart from early on, I've been 55 years now working on infection and immunity. I'm no longer doing experiments, haven't done so for a while now, and was going to retire at the end of 2019 when, of course, COVID hit. And so I was at the university, at the Wistar Institute for 7 years, back in the National University in Canberra for about 5 years, and then at St. Jude Children's Research Hospital in Memphis for 14 years full-time and then part-time, commuting backwards and forwards between University of Melbourne, where I'm in a new institute that's named after me, the Institute of Infection and Immunity, wonderful institution, brings the applied diagnostics, the virology diagnostics, bacterial, clinical medicine, together with the academic Department of Microbiology and Immunology. That synergy has been fantastic. And we've been a lead institution in Australia in the COVID-19 pandemic. And of course, I haven't been to the States for a couple of years now, not since December 2019, because of COVID-19 and because of all the travel restrictions. And I haven't been at work much either. I've been mainly sitting and doing this, talking into a computer. So it's been a very different career. From about age 35, I was really established as a medical researcher and spending a lot of time at National Institutes of Health on committees and so forth in all sorts of roles. Ended up as I was chair of immunology at St. Jude Children's Research Hospital. They also made me a member of the pediatrics department in the University of Tennessee Medical School, because we had St. Jude is a private institution, very well funded, raises $1 billion a year in public subscription, enormously wealthy. If you're interested in oncology, it's a great place to work. But it has affiliations with the University of Tennessee. And so they made me an honorary professor in the University of Tennessee Department of Pediatrics. But everyone was aware of all the jokes about how vets and how puppy dogs and little kids are much the same, and they both bite, for instance. And so I was told by the chair of pediatrics that I was very welcome to be a professor, but if he ever saw me appear on the wards with a stethoscope, they'd have me shot, which is something you take very seriously in Memphis, Tennessee, because a lot of guns around. Anyway, it's been a great time. I was reclaimed. To some extent, I was still doing some work in the veterinary area, working as a board member of an international research laboratory called the International Laboratory for Research in Animal Diseases, ILRAD, in Nairobi, Kenya, which is part of a global network of food-related research institutes. There's one in Mexico, for instance, CIMIT, the Wheat Research Institute, I think it is. And so that was satisfying. And I did that for a number of years as a board member while I was, and then of course the Nobel Prize came along. And when people worked out I was a vet, I got reclaimed by the veterinary profession. And I've been very happy to do that because I think the veterinary training gave me a wonderful grounding. It gave me a terrific grounding in pathology and infectious disease. The veterinary school I was at had been opened in the 1930s, then it closed during World War II. Australia was very threatened. Japanese troops came down as far as New Guinea, just to our north, and we had to fight them back of course, with the help of the Americans. And so a lot of things shut down. This one shut down. When it reopened after World War II, it opened with a very young senior faculty, many of whom had been recruited from a research background. A wonderful parasitologist called John Sprent, a microbiologist called John Francis. And these, I think, unusually were very research-oriented, unusually for that time. I think you'll find a lot more research-oriented people in veterinary schools now, but unusually for that time, they were very research-oriented and gave us as students the idea that this was a possibility as a career. And it is a possibility as a career. There are a number of senior vets or people with veterinary training who played very major roles, particularly in the area of infectious disease. Yoshi Kawioka in the United States, Ab Osterhaus in Rotterdam. They're top of the field in flu research. Others who've very leading immunologists, Graham Mitchell in Australia, Chris Goodnow in Australia as well, Eric Vivier in Marseille, Gabriel Beltz in Australia, a lot of top immunologists. I've trained at least one of them. And that's, I think, great to see. We do have this breadth of background. The other thing I've become very interested in, of course, because of that broad rural type background that I met first through the vet school, then through going out and working on these big sheep and cattle properties in the vacations. You know, at that stage, when I was working there, the sheep, or the cattle rather, were mustered on horseback. Now they're mustered with motorbikes because the place is so flat, or they're mustered with helicopters. So it's a very different world. So I think that interaction with environmentalism and ecological systems, the first time I heard the word ecology when I went to the vet school, that really has also given me a focus on climate change and trying to speak up about that. That hasn't been all that successful, quite frankly. We're not getting the action we need. But I think as veterinarians, firstly, we're responsible for the health of all vertebrate species except human beings. That's a pretty big responsibility. And we exercise that, of course, through our clinical activities, through our activities in wildlife and zoos. And then, of course, we're also the principal players in animal production systems. And there, again, we have a fantastic responsibility, because if you look at countries like Africa, for instance, animal agriculture is still absolutely central to feeding people and providing a decent way of life and to their culture. And of course, you've also seen the enormous expansion of, say, chicken populations in Asia to provide high-value protein. That has gone with its risks, because it's one of the ways that flu viruses jump across into us. We've been very active in public health and on the international scene. And you've got two international agencies. You've got the World Health Organization, and you've got the FAO, the Food and Agriculture, and the Organisation Internationale des Epizootiques, the OIE, both very oriented towards, obviously the FAO very oriented towards veterinary work, but also the OIE very much across the spectrum. So we have a very broad spectrum role to play in society. We're one of the great caring professions, and I don't think we should ever minimize that. That's what we do, and we help, and we serve humanity. We help with companion animals and which are so beneficial to people's mental health as well as their recreational health. And so I think, and we're also, when you think about it, health professionals, medical doctors, vets, and dentists and so forth, are often among the few people that people meet pretty regularly who are actually trained scientists. So I think we've got a role in public science communication as well. And over the last few years, I've written 7 books that have been published on science, the scientific life and so forth for lay public. And I've been active on things like Twitter and so forth. This one was the first one, the beginner's guide to running. That's me. I was a very pretty child, and it got much worse after that. That has just been translated into Spanish for the first time. And then this year I published, because I've been locked up and I've been writing a regular 800-word once a week series on COVID and viruses and immunity for our website. If you look at the Peter Doherty Institute for Infection and Immunity, there's a series called Setting It Straight. And I'm now up to number 86 of those. And that some of those, along with other stuff about our institute and so forth and COVID, was published in this book, An Insider's Plague Year. I've got a bit older than I was on that other book, so that's in our front garden. So it's been a very interesting life, and what I'd say to young people that go for it. There's all sorts of opportunity. Maybe the unfortunate thing about the scientific life is it can force you to move, and it can force, because you have to go where the funding is and where opportunity is. And of course, even a country like Australia, which has some very advanced science, but only in a much more limited number of areas. So what we do well, but we don't do everything. And so if you really want to move forward, sometimes you have to go and live somewhere else. It's good if you can train in a really good, if you want to do research, try to get to a really good basic science type institution for at least a few years, 2-3 years. Now, of course, you can do such wonderful things with cattle and sheep and humans that you couldn't do when I was starting out, because of molecular technologies that allow you to do, say, look at the genome that the mRNA expressed for everything in a single cell. I mean, you can do the most wonderful science in large animal species as you can in humans. Problem is always, of course, finding the funding. But vets, from my experience, go into all sorts of roles. They're a fairly adventurous group, I think, compared with the medics, who have a much more focused idea of what they're going to do. Some of the vets are a bit crazy, but they're often the most interesting ones. So I had people in my veterinary year who ended up running buffalo abattoirs in the Northern Territory, where they were shooting out the buffalo to get rid of mycobacterial tuberculosis, the bovine form. So people ended up in all sorts of roles: university vice chancellors, stockbrokers, entrepreneurs, all sorts of things. And of course, the veterinary practitioners who do such a great job. So I'll stop talking there. And if you've got some questions, very happy to try and answer them.
Thank you so much, Doctor. That was amazing. All of your trajectories, definitely inspiring. We have here one question from Luis Araya Sanchez. I'm going to translate right here. He wants to know your opinion on the large quantity of information that we have obtained on current years about innate immune response, and how has this helped with the understanding of the immune response as a whole?
Yes, that's a very good question. When I started out a very long time ago, we didn't know anything about the cytokines or the chemokines. And basically, the only molecules we knew that were produced as a result of infection in that early innate stage were macrophage inhibitory factor. They were experiments where people would take a supernatant fluid and inhibit the migration of macrophages, or encourage their migration through a membrane. It was discovered by John David at Harvard, I think. And in about the 1970s, the very first lymphokine was discovered, interleukin-2. And so we knew nothing about this. So I was working on the killer T cell response. We had a very, very specific assay where the killer T cells would kill virus-infected cells. So we had a specific assay that allowed us to ask questions. But beyond that, there was this whole area of what were called factors. People would look at an immune response or culture immune lymphocytes in a flask, and you would get all what they call factors, and they were trying to study them. But what actually they were mixes of different proteins, different lymphokines, chemokines, cytokines. We didn't know these existed. So what opened that whole thing out, of course, was modern molecular science and recombinant DNA technology, which allowed us progressively to identify the chemokines, cytokines, and so forth. Now, of course, as we study those, as we study them either biochemically or more often by looking, say, at mRNA and an immune response, you need computational analysis to actually try and pull together how these various interactions work. And of course, that's telling us, as we do that, that's telling us a lot about pathology, because we know that for instance, if an AIDS patient who's totally immunosuppressed, someone who is in Africa and just hasn't been able to get drugs that they should have been getting, if someone like that actually catches COVID, they may be okay. The virus grows in them, but it doesn't kill them. It's the host response that's suppressed, that's killing them. So and we know that in very severe COVID, what you need to do is, at the moment they're giving dexamethasone, but in a situation where dexamethasone doesn't work, they give them an IL-6 blocker, because that helps decrease the inflammatory response. So, and as we've gone ahead with this, people like, we've discovered the inflammasome, for instance, people like Thirumula Devikanagandhi, my colleague at St. Jude, world authority on the inflammasome. We've discovered that blocking interleukin-1, for instance, starting with mice having a knockout mouse, blocking interleukin-1 can affect various pathologies like gout. So we're starting, as a result of starting to begin to understand innate immunity, and we've still got a long way to go to work out how all this networks together. And it's like climate science. You can't just look at a bit of data. You need this computational network analysis. So we've all got informatics specialists now working with us, teams of statisticians. Same people who are doing climate modeling can easily transition into basically informatics in a medical setting. So if you've got good mathematical statistical skills, it's a really good idea to keep them up, because you can get employment in biomedical research. Biomedical people steal climate science modelers, and so do the banks and the gambling industry. So you can go to the dark side and make a lot of money. So it's a good set of skills to have. So the complexity now we deal with is massive, and that's what's so different. Firstly, when I wrote a scientific paper back in 1974, there was me and my colleague Rolf, and that was pretty much it. Now, when we write a scientific paper about a virus infection, we're likely to have 10 or 15 authors, all of whom are commanding different expertise. And we've got these big interactive groups. So if you're going to do really top level science, you need to have that interactivity and these colleagues close to you, or that you can collaborate with a distance, which of course we can. So the whole innate immunity, the emergence of, say, the NKT cells, realizing about the PAMPs and all this sort of stuff, has really changed our whole perspective on immunity. I was just lucky. There were people who were trying to work with this who really got enormously frustrated because they really couldn't pull it apart until better technologies came along. The natural killer cell, for instance, you're sort of aware of these things, but what you would do is tune them out as background. Now it's the background that matters. So enormous numbers of questions in biomedical research, and I regard veterinary research or animal related, because of course so much is done in experimental animals. Also careers for vets in laboratory animal medicine, obviously. At St. Jude, we employ a team of board certified American College of Veterinary Pathology to look at animal experiments. And of course, all drug and vaccine testing has to go through preclinical studies in animals. With COVID, for instance, initially it was only ferrets and hamsters, and then we've got various variants of the virus that grow very well in mice. So mice have come to the fore, and of course, non-human primates. So a lot of activity in all sorts of industry settings too, for the veterinary profession.
Perfect, thank you. Now I have Dr. Carlos Luna Tortos, which I will tell you that is the head of the Encounter. He helps us with the organization. So we give him the word.
Thank you very much, Professor Doherty, for accepting our invitation to this session. I think it is very important, very motivating listening to you about your scientific career. Probably rural Costa Rica is not far from rural Australia you talk about. I would like to ask a more general question. I think it is common to see that people in modern societies lack of knowledge about the principles of science, which makes them vulnerable to conspiracy theories and rejection of scientific evidence. What is your opinion about the basic knowledge or the principles of science our educational systems should seriously teach at school and high school in order to improve the basic understanding of science in the future? Thank you.
Yes, that's the question. You put your finger on the question we're all grappling with, particularly in COVID. In COVID, for instance, in Australia, in Melbourne, where I am, we have 90% of our people have accepted double vaccination. But we have a percentage of people who are hesitant because they're confused, and they're confused by a lot of the really bad information that's put out there. And we have another percentage who are actually very hostile to vaccines, who've actually been attacking people working in testing centers and have been rioting in the streets because of vaccine mandates. So how do you reach these people? It's extremely difficult. As we know, the internet has enabled it's a wonderful information system. And as we all know, if we write scientific papers or we're trying to write more generally about science and society, we can access just about anything we need to find via the internet, and we can find things we didn't know existed. I found it enormously useful writing books, but it's also a wonderful mechanism for people who have disinformation or who have really bad agendas, putting out really crazy stuff, and there's a lot of it. And it's almost impossible to refute. It's like climate change denial. It's the same sort of group of people. And so basically, we're all struggling with it. So what could we do that might be better? Well, I think in schools early on, there's two things that we live by as professionals. We live with the idea of probability and relative risk. Now, people incorporate that into their thinking re particular situations. I mean, they don't cross a street against the traffic lights because they know there's a high probability they'll be run down and killed, and there's a risk. But they don't necessarily generalize that to all their thinking. So if in some way we could get across to people the idea that probability and relative risk underlie everything that we encounter, maybe that would be a good thing. The other is the idea of skepticism. Part of the problem, as you speak out, as we speak out, for instance, and you're called professor and or doctor and all the rest of it, and even a high school teacher, it's always regarded as top-down information. We are telling people what to do. And even if you make a very big effort to make it bottom-up, to try and make it a discussion and try and get people to think things through as they go, it can still appeal to many that they're being told, they're not being involved. So how we get around that and how we get people to be able to get the idea of how science works, that it's not a top-down activity, it's an investigative activity that pulls things apart from the bottom and often is revolutionary and overturns things. It's that rejection of an authoritarian model that often gets people into these conspiracy type networks. So countering it, I think, is an enormous challenge. And I think we should all, and I think this is as true for a first-year veterinary student as it is for a dean, we should all do our utmost to try and be involved in that public communication space. And I think for the younger ones of us, particularly via social media, I now have 104,000 Twitter followers, which is, means I'm fully aware of what's going on here. I am virulently attacked and accused of mass murder because I'm advocating vaccination. There's craziness out there. They'll look at adverse events reports. When you give millions of people a vaccine, some people die. And a lot of people look at that initial adverse event report that says, well, this person was vaccinated and then died, and saying the vaccine killed them. Of course, you have to investigate all these and find out whether there's any link, and most of the time there isn't. So it's a very difficult space. But I think for young people particularly, be active on social media to some extent in trying to get good information across to people. Don't let it control your life. It can destroy you. And if you've got good visual material, I think YouTube is the most effective. And the best way to get science across to people who don't understand science is visually, either by cartoons or little movies. That works. It's much easier to have a cartoon that explains how a virus infects a cell and what it does than to try and tell people, because that's much harder for them to connect to. But visual works great.
Thank you so much for that answer. Now we have Dr. Caterina Guzman-Berry, who is a fellow researcher in our National University and the head of the biochemistry department.
Hello, everyone. Good afternoon and good morning, Dr. Doherty. It's really a pleasure to be here, and thank you so much for the organization, the students, and Dr. Carlos Luna, that I know he has been doing this work for a very long time. Very inspiring in your talk. Thank you so much. And as Nicole mentioned, well, I'm not that young as the students. I'm actually a biochemistry teacher. And actually my training is in human clinical microbiology. But I got really lucky to work at the vet school at the Universidad Nacional, and I'm really having fun there. And actually, my question is related to the question that Dr. Luna just posed. And it's also related to what Dr. Garza, I think he's from Mexico, has also commented on the chat. I have written it down, so let's see if it's clear. It's related to public science communication and engagement. And as a clinical microbiologist working with veterinarians, I realized the importance of working as a team. The work is so complex that we can only try to grasp the reality by doing it each other. So I would like to ask you if you have any advice or suggestions on how to improve communication in and outside academia regarding the importance of a One Health approach and the role that countries such as Costa Rica, with the limited resources, but huge biodiversity and human resources, can contribute to the advancement of emerging infectious diseases.
Yes, I mean, it's a little bit the question we approached before, but I think some things that are very important as principles. One is that when you're talking to people, either whether it's in your clinic or you're asked to give a talk, say someone may know you know something about, say, COVID or something, they ask you to talk to a local community group or on a local volunteer type radio or anything like that. Try and find very simple examples and make sure you never ever talk down to people. Don't come across as I'm the authority, I know you don't, sort of thing. Try and relate to them and their experience. The other thing is, you're giving a public talk, never, even no matter how silly a question may be, never humiliate the questioner. Because if you do that, and talkback radio is another way that this happens. If you do that, even though everyone listening may agree with you that this person's crazy asking this stupid question, if you humiliate them by really talking down to them, you will lose your audience immediately, because people hate that. They hate people talking down to people who don't have an adequate understanding. The other thing I think is great, and with Costa Rica, as you say, with your enormous biodiversity and your wonderful natural environment, is at least for those people who have their own financial resources to have time to spend doing something other than earning a living. I think it's very good, and you could do this through organizational structures to do everything possible to promote the idea of citizen science, because that really engages people in the broader community, and it will engage people who have no scientific training or no interest in science as such, as bird watchers, or in monitoring the health of rivers for pollution, or in monitoring what's happening on the beaches. I mean, a lot of people who have no scientific training can name enormous numbers of plants and give you the Latin names because they're gardeners and they love plants. So the more you can engage with those sorts of communities, and bird watching is the classic one because so many people are involved in it, and say bird counts, the backyard bird count that the Audubon Society runs in the North America. We have the same thing here. And the more you can engage people in that activity and then get them to actually collect data that feeds back to scientific professionals. And this is what happens with the birds. You know, if you report back through the backyard bird count, then that's handled in programs that are designed by wildlife ecologists and zoologists. to actually get useful data out of it. And it all goes up online. I think it's Columbia University has this whole thing about birds across the planet, essentially, that a lot of it feeds in from lay, untrained observers. They don't need a science training. They just need to be able to recognize the bird. And they become involved in science without realizing it. But you can actually seduce people into thinking scientifically by involving in that. So I think that's one mechanism we can use is the inclination, especially at least in our society, as many retired people have a lot of time on their hands and have adequate income to actually get involved at that level, and young kids as well. And of course, school teachers often try to do this with their children, involve them much more. Unfortunately, with all the stresses of adolescence and so forth, that often gets forgotten. So we need to reintroduce them to it a little bit later in life.
Thank you so much. We have some comments from Juan Garza, all the way from Mexico. He says that there is a vision that we as veterinarians should be responsible for areas such as One Health. However, the pandemic has provoked that we work in a transdisciplinary way. The leadership of the veterinarians is supported by our wide, transversal, multi-sectoral vision. We should encourage more multidisciplinary work, such as we have done. He congratulates you for your career and thanks you for taking some of your time to invite us to follow your steps. He gives you best wishes, hopes to see you soon in Mexico, and he tells us that he was a former dean back in the 70s. Okay. We also have another question by Luis Araya. He says that the new century has brought huge interests about the immune response in the geriatric patient, third age. So he wanted to know if you could comment on changes that the T cells suffer as we age.
Yes, I just comment a bit on the One Health thing. I've been involved in this right from the beginning of the One Health movement, really, or from formally it coming together. It has largely been driven actually by veterinarians, particularly by vets who are interested in infectious disease, often working in the medical area. So there's a whole component there of the infectious disease component, which you see reflected in some of the big One Health meetings. But it also encompasses what we used to call comparative medicine. You know, an example I use is McConnell at University of Calgary Vet School, who's doing research on antler velvet in deer, because, you know, they shed their antlers and then regenerate them. So if you can understand how antler velvet works, does that help us understand human regenerative medicine? So the One Health thinking, which is multidisciplinary across the spectrum, and if you wanted a vet school that works very closely with the medical school, one is the University of Calgary in Canada. It's kind of a tiny vet school, a bijou vet school, but the vets and the doctors teach across both faculties. So I think that's a wonderful model, actually, if you can get it to work. But of course, we always know that in universities, the structures are such that it's not easy to do that. But the more we can do it, the better. And so the second thing about geriatrics, traditionally, clinical immunology was very much dominated by two specialties: rheumatologists who deal with autoimmune disease and neurologists who are dealing with multiple sclerosis, again, seen as an autoimmune disease, which it undoubtedly is because you can suppress it by suppressing the T cells. So they were the two disciplines that did a lot of work in immunology. But the two disciplines that we really needed to be doing a lot of work weren't heavily involved. One was surprisingly pediatrics. There weren't a lot of people in pediatrics who are really interested in immunology. That's changed. And the other is geriatrics, gerontology, which is a kind of a late emerging specialty in many places. Now, with our modern technologies, and my colleague Catherine Kizierska, for instance, who sort of trained with me and now is doing human T cell responses, B cell responses, that's changing. And we're starting to look at the immune cells themselves in people, because previously, there wasn't a whole lot you could do with immune cells, particularly from a baby or from an elderly person. You didn't want to bleed them all that much and all the rest of it. But now you can get cells fairly readily. older patients, particularly older people, if they're in retirement homes and so forth, are really quite happy to be involved in research studies and to be signed up for observational clinical trials where you can actually look. And now we can look into the T cell from a young adult, from a geriatric patient, from a young child. We can actually look into single T cells and see what's going on there. We can look at their B cell responses quantitatively. We can look at their telomeres and all the rest of it. So the whole thing is starting to move, but it's been slow, and it's still not a major focus in geriatrics. We need many more people and more funding. We've had the National Institutes of Health have had an Institute of Aging, but in immunology, it's really, I think, really starting to emerge now, and how we try to counter this effect of aging. We do know from early mouse studies and so forth that we're losing immune repertoire as we age. We lose what's called public TCR specificity and so forth. But I think it's a field that now is starting to go places. And I think it's very central to protecting the elderly from infection, because of course the people who are dying from COVID are mainly, and even double vaccinated, what we would describe as the frail elderly.
Thank you very much. We have another one from Juan Rivera. He greets us from Colombia, and he says that in your experience, which are the basic principles of immunology that we are not taking into account in the moment of the private practice with small animals that affect the recuperation and prognostics of our patients?
Gosh, I really don't know the answer to that, because I'm not really involved with small animal practice and the realities, the business realities of small animal practice. But I know that, of course, now when I started in veterinary medicine, there was no subspecialty structure at all. I mean, you know, you didn't have specialists in laboratory medicine and all the rest of it that you could refer to, and it just wasn't there. In fact, clinical medicine was only really developing that. Human medicine was only really developing that formal structure. So I think the assays that are easy to do in patient side, for instance, the lateral flow antibody tests that we're now using, and lateral flow antigen tests, where you can just take a drop of blood, put it on a slide, and then watch it develop and read it off with some sort of reader or by eye. I would think those types of assays are appropriate it could find a use in the clinic. But, you know, veterinary medicine, at least in my day, didn't focus at all on the declining immune system in aging animals. I mean, you know, that really wasn't, geriatrics wasn't really a developed area, whereas, of course, now people have a very different approach to this sort of question. So I think simple tests that you can do easily and that can be marketed as commodity kit forms would obviously be appropriate, because they're useful. I know they can be used in medical practice, so I imagine veterinary practice is the same. What their actual applications would be. I mean, for instance, I mean, it would be possible, for instance, to have some sort of diagnostic kit which gave you a diagnostic for all the autoimmune type diseases, which of course can develop in domestic and pet animals just as they can in humans. So it's an area though that probably could be considerably developed. The availability of easy to use kits that would be useful for monitoring, particularly degenerative disease type situations. But you need research and people willing to be entrepreneurial and make some money out of it, which might be a way for a young veterinary scientist to think.
Awesome, thank you. We have another one from Jose Vargas. He says, what would you advise a veterinary student, or any student in general, to develop and promote a questioning mind in a world where there's so much information at your fingertips?
Yes, that's a it's complexity, isn't it, that we're facing? We have this information overload. So what you have to do, of course, is focus, and that's kind of the sort of personality I have. I like to ask a question and then go in depth on that question. And the internet helps us enormously with that because we can get all sorts of information very quickly. The other thing that helps enormously with questioning is to have a friend who's questioning and to be in a group that's questioning. Some of the great science that's been done has really been done by people talking to each other. There's a little book on the life of Francis Crick, Watson and Crick, the two guys who first discovered what DNA is for. They published this in a letter to Nature, one of these ones I described, where you've only got, say, 3 or 4 pieces of data, and you've got 1,000 or 1,500 words, and they described, how they worked out the structure of DNA. And in the very last sentence, they have a sentence that begins, "It has not escaped our attention that," and they basically say, we've explained inheritance, and it's sort of this little modest sentence right at the end, typically British, which has this enormous punchline in it. So, but the secret to Watson and Crick was that they just talked all the time, and they threw ideas backwards and forwards and pulled ideas apart, and then eventually constructed that model of DNA. If you watch that movie, there was a little BBC movie made called The Double Helix that sort of goes into this. So they were just asking questions. Crick was a physicist, so he understood crystallography type orientation. So he understood something of molecular structure. Watson was a biologist who'd done a PhD in phage genetics with Sally Luria in Purdue, I think it is. And so these two very different people from different backgrounds, that's also very important. Just talk to each other all the time. After Watson and Crick broke up, Watson went off and eventually became president of the Cold Spring Harbor Lab, which is really the home of molecular biology. It's where it really started with phage experiments back in the 1940s after the war. Actually started by a physicist called Max Delbruck. There's a picture of Watson at Cold Spring Harbor actually working as a waiter at the tables, which was a vacation job when they used the American academics who had 9 months' salaries all came to Cold Spring Harbor in the summer and did experiments with phage. You grow bacteria, viruses that grow in bacteria, grow them on a plate, do an experiment overnight, drink beer, they'll set it up that morning, drink beer that night, come back and read the experimental results the next day. And that's where molecular biology started. So, and then Crick, after he stopped talking to Watson, started talking to Sidney Brenner, who got the Nobel Prize for differentiation, again in Cambridge. And so that they talk backwards and forwards all the time. So talking to people is great, but going in depth on and focusing. One of the things that sometimes we have a problem with when we do a very broad training like veterinary medicine is actual focus. But if you're going to do something in science, you've got to be focused down on it. And you've got to ask specific questions. Doesn't mean you're stuck with that sort of question all your life. I mean, one of my absolutely best colleagues, I mentioned it before, Thiru Devikananda, has taken apart the inflammasome in autoimmune type conditions, but her training is in botany. So, you can go across barriers and so forth, but you have to be in a situation where you can ask those questions and go in depth, which is what actually a PhD training should do for you in a good lab.
Perfect. Thank you so much. I have this next question is mine. I know you are passionate about history. So I wanted to know if you have a favorite civilization and which one would it be?
A favorite what?
A favorite civilization.
Civilization.
Civilization, well, yes.
I guess my fascination has really been with the world with modern science. So basically, I'd say I'm sort of fascinated by what really began in the 17th century, even in the 15th century in Europe, after the church, the Catholic Church, had really for centuries controlled intellectual activity and had limited inquiry to subjects that didn't contradict their teaching, because it was a very authoritarian organization. And so in the, I think it's the 1600s, a group of students broke away. I mean, the Catholic Church controlled the universities pretty much. A group of students broke away from the university structure and started their own university in Padua. And then a similar movement in Bologna. And these were the universities that first started doing dissections again. You couldn't do dissections. That was regarded as witchcraft or so forth. So they started to do dissections again. And people like Harvey from England, a surgeon from England, came to Padua to do dissections on eels and fish and dogs to work out the circulation of the blood. You know, they were working from the ideas of Galen in ancient Rome, that the blood was created in the heart and flowed to the liver or something. I mean, nobody was talking about the heart as a pump, which is really extraordinary when you think that every time you kill an animal and cut its throat, it pumps blood everywhere like that. So people didn't make that connection. They were not thinking in a way that emphasized curiosity, because actually it was dangerous to do that. You could be punished severely or burned at the stake or whatever. So you didn't do that. You didn't think about curiosity. So Padua really started to open out the whole of, by dissecting again, and people came together illegally to watch human dissections and to see what the body was made of. And Malpighi, for instance, in Padua, I think it was, or Bologna, Padua, I think, started to study embryology by studying chick embryos. Nobody had done that. So he started to understand embryology. He also described the capillaries for the first time. So it was this age of curiosity. And then Leeuwenhoek and so forth a little bit later in the Netherlands, making simple microscopes. And it started in England, at least, which is what I know best, with a group of people coming together and influenced by the thinking of the former Lord Chancellor of England, the Treasurer of England, Francis Bacon, who was fired actually, but managed not to be executed, as people who had those sort of positions often were at that time, and went and thought about knowledge and natural philosophy. And he developed the idea that you couldn't just make statements about science saying this is the way it is. What you had to do was actually make the observations. You had to do the measurements. And then, so that led to a sort of philosophy where people said, well, yes, we need to do this. We need to look at things, look at the thing itself. And so they came together to form a little club, which became the Royal Society of London, the first scientific academy in the English-speaking world. It had all sorts of people who weren't scientists at the beginning. It had Christopher Wren, the architect. It had Newton, the scientist, Hooke, the scientist. It had Pepys, the diarist, who was Secretary of the Navy. They all came together to form this club. And it was given a charter by King Charles II in 1645 or something like that. And he also gave them a book made of vellum, calf skin, which if you're elected to the Royal Society of London, which is the oldest academy, scientific academy in the English-speaking world, and serves as the National and British Commonwealth Academy of Science, you sign this vellum book. So I think it's that little beginning. And then the Royal Society set up these rules, and Bacon insisted everything had to be made available. If you made observations, you had to make them available to everyone else. So Bacon set up the rules that it had to be, not Bacon, the Royal Society set up the rules that everything has to be published and it has to be reviewed before publication. And this begins the publication of science. And the very first scientific journal in English language is Philosophical Transactions of the Royal Society of London, now a review journal. But you can go back online and read articles that were published in 1650 or so in the Transactions, the Philtrans. So it's this beginning that really, starting in the 17th century, has given us the society we know today, that has so transformed society by those simple rules make the observations, do the work, write it up, think about it, publish it after peer review. That has transformed our world. That's the world we're in today. Of course, it's given us good things and bad things. It's given us really a somewhat overpopulated planet. It's given us climate change due to anthropogenic warming and so forth. So it has its pluses and minuses. And really the only way forward, I think, is, apart from trying to change people's behavior a bit, is to use the mechanisms of science and technology to try and find solutions to the problems facing us. So it's that period that fascinates me, from the 17th century on, quite frankly, and particularly the 19th century, when all sorts of incredible things were happening. And then our own era. I mean, science has been transformed totally in my lifetime.
Thank you so much. I don't see any more questions. I don't know if anyone would like to raise their hand. Send another one by the chat. Oh, Dr. Carlos Luna.
On mute.
Yeah, you're on mute, doctor. Dr. Luna, we can't hear you.
Right now.
There you go.
It's me again. Thank you very much.
I'm going to have to go off after this, but,
sorry?
I'll have to go off after this. I've got another thing coming up. Yeah.
Yes, just to tell you, this is the 9th year we celebrate this meeting with our students in Costa Rica and at our university. And from the very beginning, I was completely convinced that this should be in their hands, or the whole organization, because I really identified a lot with what you say, that the science has to be very cooperative, and it shouldn't be from top to down, but very horizontal. And I really like a lot your words and your philosophy, because I'm really convinced that if in science, if we are working together and sharing the knowledge and communicate at the same level, the science is the way it goes up and advances and improves. And I really appreciate a lot your participation in this session and in our meeting. Thank you a lot.
Well, thank you very much. And I think that's a wonderful point to end on, that basically the science depends on a collaboration between experience and youthful enthusiasm and novel ways of thinking. And I think if you read some of the stories of scientific careers, which you can actually do on the Nobel Prize website, they have the stories, or they have the actual biographical details and also details of the science that have been given. And particularly young women might enjoy interacting, say, with the Nobel lectures and the Nobel biographies of, say, Jennifer Doudna and Emmanuel Charpentier, who got the Nobel Prize, I think, last year or the year before for their molecular genetic studies. So I think there are great examples out there that are worth looking at and reading about. And in some ways, science has been like this for a long time. And it's been my great privilege to continue working with young people and smart people. And that's one of the wonderful things about science. Though as a senior scientist, you have to make sure that those young people get due credit for what they've done. So thanks very much. And it's been a pleasure to speak to you. And good luck to everyone in your future careers and whatever you decide to do.
Thank you so much, Doctor, for your time and words.
Okay. Thank you very much.
Thank you. You have a good day.
Yeah, I say bye.
Muchas gracias a todos los participantes.
Gracias. Adiós.
Tengan buena tarde.
Buenas tardes.
Muchas gracias.
Hasta luego.
Más bien, muchas gracias a ustedes. Muchos éxitos.
Muchísimas gracias.
Buena vida, gracias.